thank you to the organizers for the chance to give a presentation today. It's kind of exciting for me to be here at BIPM. A little bit of story about me. I actually was a NIST research scientist for several years and in a radioactivity group, and shared an office with Brian Zimmerman 20 years ago. And it was actually during that time that I was introduced to this idea of alpha particle radionuclide therapy for cancer by a physician scientist at University of Maryland in Baltimore, Maryland, in the United States. He convinced me that, that we could use these alpha particle emitters and that they could potentially be transformative for cancer patients. In our first experiments looked really fantastic. And so that leads me to a 20-year story to standing here today to talk a little bit about lead-203 and lead-212. It was difficult to follow the presentations that came before me, but I'm gonna drill down into some of the decision making that led us to this idea that this elementally matched pair of isotopes could really be transformative for cancer patient care. So this is a little bit of a forward-looking statement, but I think that we're seeing this momentum in oncology today that is telling us that, this radiopharmaceuticals, and in particular, alpha particle radionuclide therapy for cancer can be, another pillar in oncology. Chemotherapies, radiation therapies, antibody drug conjugates, immunotherapies are all pillars of cancer patient care. But there are certain aspects of this that have been brought up in some of the presentations that we've seen today. They're molecularly targeted. So we're delivering, the cytotoxic dose specifically to the tumor microenvironment. And then with alpha particle radionuclide therapy, really at the cellular level, we talked about the differences between alpha particles and beta particles, and I'll talk a little bit more about that. The idea of being able to use imaging to select patients that could benefit from therapy was unique to oncology for this sort of theranostic paradigm. I like to call it image-guided radionuclide therapy for cancer. The idea of being able to be able to make a decision about whether or not a patient can or might benefit from the therapy is unique to oncology. And I think that that is part of the engine that is fueling, or part of the fuel, that's powering the engine that is bringing these new therapies to cancer and leading to the presentations such as our first presentation today from South Africa. The potential certainly for alpha particle radionuclide therapy for cancer as being an outpatient friendly clinical scenario for patients is definitely something that we're exploring, and it looks as though that this might be actually possible, and that makes it better for the patient. So if you rearrange your thinking into what is this going to be like in the patient care setting, then certainly the supply chain that we've talked about is really important, and I'll talk a little bit more about that. All of this put together makes this for, a unique business opportunity, right? And so that's really important for fueling this economic engine to allow the manufacturing and distribution of these new radiopharmaceuticals and alpha particle therapies for cancer patient care. So this differences in alpha particles versus beta particles have been brought up in the presentations prior. Beautiful video from Dr. Napoli. And so the idea here is that the alpha particle, 7,000 times the mass of a beta particle, much higher energy. It has the potential to deliver cytotoxic dose to cancer cells in a very short range, which potentially makes for the less potential for off-target damage to normal and healthy tissues. Now, I would like to stop there, though, and talk a little bit about the molecule also does matter, and the chelator matters. So, and I'll talk a little bit about how I chose lead-212, how we chose lead-212 as the radioisotope for alpha particle therapy, along with the importance of the chelator and the delivery vehicle for making sure that you get radiation dose to the tumor microenvironment and minimize it to other parts of the body. That kind of leads to just a brief story of how we got to where we are today. So I was a co-founder. I'm joined with a co-founder here, Francis Johnson, and our director of isotope manufacturing, We started the company, basically at the kitchen table. And we've grown now to a company that is delivering radiopharmaceutical therapies globally. This is partly driven by the data that's been generated, and we saw some of that earlier today about how transformative the radionuclide therapy, and in particular alpha particle therapy, could be for cancer patients. And so this is, the economic engine is really important for this, and I'm proud to have taken this from an idea with a physician scientist at the University of Maryland to where we have it today. From a clinical perspective, we're a company that now has two agents that are in clinical trials in the United States, and we also have an active expanded access program. Some of that data has been published at the EANM with our work with Fortis Hospital in New Delhi in India. We have a program for neuroendocrine tumors that I'm going to highlight today, in particular to give you some ideas about the potency of alpha particle therapy compared to beta particle therapy. And I'll show some data in the preclinical setting and the clinical setting that helps you to understand the differences that we're seeing there. I will also talk a little bit about how we can use lead-203 for this patient selection and imaging and dosimetry that's going to be so important as we bring new agents into the clinic. And you see several that we have here that are in the preclinical to just about entering clinical setting, and how important that lead-203 elementally matched isotope can be for early drug development as well as clinical development. So let's talk a little bit about what I think about lead-212 and what led to this decision to choose lead-212 as a radioisotope for alpha particle radionuclide therapy. This is sort of a picture that's kind of the history of radionuclide therapy and theranostics, right? We all know about iodine-131. It's been used for decades very effectively for treating thyroid cancer. That's also imageable, so it had some potential to be able to use be used as an imaging agent to select patients for cancer therapy, potentially for dosimetry. But over time, we realized that we could start taking delivery vehicles, antibodies to start with, right? We looked at antibodies because we know we can select them with very high affinity and selectivity for a particular molecular target. But over time, we started to recognize that peptides and peptide-like small molecules were probably the best delivery vehicle. So the idea there was to deliver radiation very quickly to the tumor microenvironment and have peptides and peptide-like small molecules that we could engineer, using medicinal chemistry to target the tumor quickly, but then have the residual dose, wash out of the body very quickly, usually, via renal clearance. And as we selected those, we just in thinking about what would be the right isotope for that, we chose lead-212 because the half-life of lead-212, was much more well-matched to these peptides and peptide-like small molecules that, unlike antibodies that have circulating half-lives that are very long, these have short biological half-lives, so clearing the blood in an hour or so. And so, a shorter half-life seemed to make more sense. The other thing that I'll talk about a little bit more is the supply chain and the ability to be able to create a supply chain management system that would allow you to be able to ensure that you had local regional delivery that had a backup supply from somewhere else. So, rather than the idea of having a single continental source that would deliver, from one single site, to have a network of sites similar to what we did with F-18 FTG, in the late 80s and early 90s, and it proliferated into radiopharmaceutical manufacturing sites around continentally, both in the United States and around the world, to be able to deliver a 2-hour half-life to the patient bedside. The other thing about lead-212 that I really liked, and I was convinced that at some point, as Dr. Vorster talked about earlier today, physicians were telling us as we went around the country talking to them about what would be important aspects of a radionuclide therapy for cancer, and the idea of having an imaging surrogate that they could truly use to be able to develop dosimetry plans for patients. particularly in the early stages of clinical development, would be an ideal counterpart to an alpha particle radionuclide therapy for cancer. And lead-212, lead-203 in the current time are the only practical elementally matched isotopes that can be used in this context. So we can not only use lead-203 very effectively in the preclinical setting, so you see a mouse image that we obtained with our gamma eye instrument, which we find very useful for. Secondly, when we get to the early, clinical development phase and early clinical trials, we can use this lead-203 biologically innocuous imaging surrogate to be able to make detailed determinations of the biodistribution and PK properties of the molecule. And because lead-203 and lead-212 are elementally identical, we can be really much more confident that the predictions that we make with the identical molecule will be predictive of what we will see with the alpha particle therapy with lead-212. So I'm going to show some examples here, or an example, I guess, preclinical data that demonstrates the potential for alpha particle therapy relative to beta particle therapies. And then I'm going to talk in the end, just show a couple slides about, what are the complications of these alpha particle therapies that lead to, I think this meeting today and trying to develop a better understanding of what metrology could be done to improve on our understanding. So this is a VMT AlphaNet is a somatostatin receptor targeted peptide that's been modified in a proprietary way to improve on its PK properties. We were granted a fast track designation by the US FDA, and it's now in phase 1 clinical trials in the US. And I'm going to also show some compassionate use data in the clinical setting of the therapeutic that shows the power of this agent. In the preclinical setting, this slide is just really meant to help you to understand the potential superior efficacy that can be achieved using alpha particle radionuclide therapy for cancer relative to beta particles. So if you look in the upper left-hand corner here, you can see the control study. This is a somatostatin receptor positive tumor cell line that's been inoculated into mice, and these tumors grow very rapidly in the absence of treatment. On the right, you see lutetium-177 dotatate. This is a FDA-approved therapy in the United States. It's been used for several years, and you can see why this was approved, right? So you can see that, it controls the tumors. It improves on progression-free survival by almost a factor of 3 over this period of time. But when we administer either a single dose, if you look on the bottom part of the screen here, either a single dose of 120 microcuries or 4.4 megabecquerels, we see 100% complete responses. Or if we use a fractionated approach to this at 4 doses of roughly 1 megabecquerel, that we also see this incredible response. So the difference in potency in alpha particle radionuclide therapy versus beta particle therapy are pretty evident here. So that the next step, then you go from the preclinical setting to the clinical setting. And the idea then is, do the PK properties of this molecule once we get to the clinical setting, and this is another example how we can use lead-203 imaging to help us to understand that. So we see rapid tumor accumulation here at an hour post-administration. To be able to see liver metastases by a SPECT whole body image is pretty remarkable and shows very precise tumor targeting and rapid renal clearance of the residual dose, exactly the way that we had developed this molecule. And then just as exciting, maybe more exciting, 21 hours post-administration, we see excellent retention. So this tells us that over, the 2 half-lives, or roughly, 85% of the lead-212 alpha particles will be emitted in the tumor microenvironment over that 24 hours after following the dose administration. This tells us that we're getting an effective tumor dose to this patient, and this patient, then the physician would likely decide was a good candidate for this alpha particle therapy. So we've learned more about the potential for imaging over the last year or so, and these are some images of a different patient. So on the left-hand side there, the lead-203 SPECT imaging of a patient showing concordance with the PET-CT gold standard gallium-68 net spot imaging that's done in the center where this patient was imaged, followed by, once again, here we see this nice concordance with the PET-CT imaging on the top left there with those two images at 1.5 hours, and then nice retention of the tumor dose in the tumors at 24 hours. Now, what we've found since that time, and this was the first image of lead-212 SPECT-CT ever in a human patient. This is a VMT AlphaNet lead-212 image that was done by our colleagues at Technical University of Dresden in Germany, where you see beautiful concordance between the lead-212 SPECT imaging with the gallium-68 net spot that was done to select this patient for therapy, and then nice, once again, retention of the lead-212 dose in the tumor at 19.5 hours post-administration of the drug. So this is another scenario where now we've got a second tool that can be used. We can image the patient before the therapy is administered using the lead-203, be confident about the biodistribution and tumor accumulation and retention and other organ washout about the agent, prepare a dosimetry plan that would be based on this data that would help us to deliver the appropriate dose of lead-212, and then post the administration of the lead-212, we can actually do a confirmatory scan to help the physician to know that it looks like this drug was actually retained in the tumor and delivered the dose that we expected. This is an example of the power of alpha particle radionuclide therapy in a clinical patient. So this is a patient that was selected for compassionate use in cortisol crisis. Here you can see the ACTH levels were abnormally high, and this patient was really sick. But this image that you see on the left-hand side of the screen there is about 6 weeks before the administration of the radiopharmaceutical therapy. The dose, or the image that you see in the middle there, both PET-CT and the SPECT-CT show, a very nice response to a first dose at levels that were about where we are starting our clinical trials in our dose escalation study. So relatively low dose, really nice response. And after 3 doses, we're seeing this patient's therapy is almost completely resolved. And the really nice part about this was this patient was really sick. And she went back to work, and she went on holiday to Hawaii with her family following these therapies. So a really nice story, but mostly here just to help you to understand that these therapies, this would be unusual for, the standard of care Lutathera, lutetium-177 therapy for a patient in this setting. So I think that I hope that I've given you some insights on the potential for these alpha particle therapies and why there's this momentum in the clinical setting, to provide these on a regular basis to patients. And one of the things that I really liked about lead-212 also is that we reduce the supply chain to, chemistry and chromatography, right? So we can obtain thorium-228 around the world. We have a great relationship with the U.S. Department of Energy, and there's more sources around the world that we've identified for the thorium-228. That means that we can have, because thorium-228 has a 2-year half-life, that allows us to be able to inventory that and the radium-224 that comes after it in a way that allows us to, establish this local regional manufacturing and distribution that always ensures that we've got an inventory at least one of our sites that can make sure that the patient gets the dose in their treatment regimen that's necessary. And we've seen, issues over the past few years, and nuclear medicine knows this. A single site of manufacturing goes down and we can't get lutetium, or we can't get a lutetium, or we can't get technetium-99m for months and months and months. That means that a patient who is in their treatment regimen has to make, and their physician need to make some really pretty tough decisions about what to do until that supply chain can be reestablished. So we have worked over the years to ensure that we've got a supply chain that's quite robust. So the last 2 slides are really, what is the issue about these alpha particle therapies, and why are they challenging? I happen to be part of the radium-223 team at NIST when I was there, and worked on radium-223 standardization when I was at NIST. And I think that each one of the alpha emitters that we're talking about here, other than the terbium isotopes that were mentioned, they all have a decay chain, right? So there's a series of radionuclides. So what we have is a moving target in metrology. And that turns out to be a challenge for making measurements on a timely fashion, particularly, at the quality control release, right? So when you've done a purification, now you've got decay and ingrowth going on at the same time. And so that presents some challenge. And so I'm really excited that the metrology community is really taking aim at this. And we've really appreciated working with Brian and Jeff Cessna and the other, and Dr. Karam at NIST, and Sean Collins, who will speak at the NPL, to develop this unbroken chain of measurements with stated uncertainties all the way to the clinical setting and the dose calibrator in the clinic that can help us to ensure that what we have, that what we are administering to the patient is certainly within a stated uncertainty, what we thought it was. And actinium-225, another one of these isotopes that has this complex decay chain, these therapies, can be highly effective for cancer patient care. And so it's also critical that we're positive about what we're administering to the patients, and that's where the metrology community comes in. And I'll stop there. I hope that I've convinced you that this can be a really potent therapy. I really believe that lead-203 and lead-212 as an image-guided pair have great potential and are powerful tools. But these decay chains present some, potential challenges to the metrology community, and we're happy to help, and we appreciate the effort. And thank you for your time and attention.